Yeah, hi everyone. I'm Haihan Zhang. I'm a recent PhD graduate from Department of Biostat, and I'm currently a postdoc research fellow in the Department of Dermatology. Yeah, it is my great honor to be here to have the chance to talk about my work on keyword fine mapping approaches to prioritize potentially causal signals underlying binary traits. Yeah, so first I just want to have a quick introduction of TWAS, which means the transcriptome-wide association study. So this is very similar to the more well-known GWAS study, which usually tested the association between the genotype and the trait of interest. So here for traditional TWAS, we are trying to set up the association test between the transcriptome and the trait of interest. Since most of the GWAS panel do not have a known gene expression level, so the first step of the TWAS will be the prediction model setting up. So on the left part of this slide, you can see we do need a reference panel, which is usually just the EQTL panel made from different tissues and covering hundreds or thousands of genes. And we do need to have the known genotypes and the known gene expression level from this reference panel, and using usually linear model or more fancy models to set up the prediction models to get the SNP weights for each gene, so we can further predict the gene expression level in an independent GWAS panel. So after we got the prediction model, we just plug in this prediction model in another GWAS panel where we do have known genotypes. So with the known genotype and the prediction model, we can generate the imputed transcriptome, which is also known as the predicted gene expression level. Then we set up the association test between the predicted gene expression and the trait of interest. So yeah, just to be clear, so the trait here can be either, continuous outcomes or binary. So my work here more focused on binary outcomes. Yeah, so in advance to the TWAS, since TWAS is a marginal association test between a single gene and a trait of interest, so it will ignore the pleiotropic effect, which means the single genetic variants can independently influence both the gene expression level and the trait of the phenotype. And also, the marginal association test will ignore the expression correlation among nearby genes, usually induced by the LD structure. So in addition to TWAS, now we are focusing on developing methods for TWAS fine mapping. This is also like a terminology borrowed from the GWAS fine mapping, so which means we can jointly model multiple genes at the same time to identify the specific gene or the set of genes at a given locus that are potentially causally responsible for the association with a trait. Yeah, so also we know that most of the diseases are usually complex, and multiple tissues or multiple conditions can be involved for a specific disease or for a group of diseases. So people started to developing methods for multi-tissue TWAS. There are two well-known, more popular methods in this field, which one is called multi-skin, which is an extension of the previous skin. And the other one is called the UTMOST. These two are more known methods and more developed methods in this field of multi-tissue TWAS. And in addition to multi-tissue TWAS, people also got the interesting to developing methods for multi-tissue TWAS fine mapping, which, as I mentioned, can jointly model multiple genes at the same time. And now for multi-tissue TWAS fine mapping, we can even jointly model multiple genes across multiple tissues. So here is the outline for my talk today. So it will be divided into two parts. So first, I want to talk about Fabio. This is a statistical method on single tissue TWAS fine mapping, specially designed for binary outcomes. And based on that, we further developed the mFabio. This is also a statistical method that is designed for multiple tissue TWAS fine mapping and also specifically for binary outcomes. Yeah, so first I want to briefly talk about the motivations of why we are trying to develop the single tissue TWAS fine mapping method. So first, most of the existing TWAS fine mapping methods were primarily designed to model quantitative traits, which means they are continuous outcomes. However, even though linear regression is a very popular approximation way to logistic regression in TWAS and also in GWAS settings. However, unlike the SNP weights in GWAS, the GTEx, which means the gene expression level effect sizes observed in TWAS analysis can usually be observed relatively large. And also, existing TWAS fine mapping methods only consider the correlation among gene expression level within locus, resolution, which means usually they can be only applied to LD loci, which only covers like up to like 100 genes, usually much smaller than that. And we do know that the cis-SNPs of the genes that are outside of the same LD regions may also have a relatively higher correlations. So this will result in potentially correlation among gene expression levels across multiple LD loci. So here we present Fabio. This is a fine mapping of causal genes for binary outcomes. So generally speaking, Fabio follows the traditional TWAS fine mapping ways, which is a two-step manner. So in the first step, we do need another EQTL reference panel where we do have known genotypes and the gene expression levels, so we can set up the gene prediction model. And after that, we plug in this gene prediction model to another GWAS panel where we only have genotype, so we will got the imputed gene expression level for that GWAS study panel. And afterwards, we mainly apply the probability link for binary outcomes, so we can more accurately to capture the variance among those genes, especially when they are associated with a binary outcome. And at the end, this is a Bayesian method, so we are calculating the posterior inclusion probability for each gene to serve as an indicator of whether or not a gene is potentially causal for this trait of interest. So we first using extensive simulations to evaluate our methods against the other existing methods. So we are using the UK Biobank to serve as a GWAS panel, and we use the GWAS data to serve as the EQTL panel. And we first simulated the gene expression in both of the EQTL reference panel and the GWAS study panel using the real genotypes. And there are two key tuning parameters in the simulation. One is called PBE1. This stands for the proportion of gene expression variance explained by causal cis-SNPs for each gene. And the other key parameter we are using here is the number of causal SNPs for each gene. So things for the EQTL reference panel, we only need the real genotype and the imputed, the simulated gene expression level. However, for the GWAS study panel, we also need to simulate the binary outcomes to try to showcase the advantage of our method against the other linear regression model-based methods. So here we use the logistic regression to set up the simulation and got the binary simulated outcomes. Again, here we have three key parameters. One is case control ratio, and the second one is the number of causal genes for each trait. And the last one is PBE2, which stands for the proportion of phenotype variance explained by each causal gene. So here we first trying to show that Fabio can successfully control false signals in non-simulations. Here we compared Fabio to Focus and Fox, which are two popular methods in the field and also existing method in the field of TWAS fine mapping back to the time, I believe 2022, when we started to preparing for this manuscript. However, there are more existing methods like GIFT and CTWAS developed more recently, and they also have like a relatively good performance in the TWAS file mapping. However, we did include the comparison of Fabio and those more newly developed method specifically for the case of binary outcomes. And we also observed that Fabio still can outperform them. So although these results are not covered here in my presentation today. Yeah, so again, back to that time, we only compare Fabio to Focus and Fox, and we observed that in addition to the non-simulations, Fabio can also achieve the highest power in multiple alternative simulations. And furthermore, we move from the simulation to the real data application. So again, we are using UK Biobank data to serve as a GWAS study panel here for real data. And we picked 6 binary disease outcome here, and they are asthma, breast cancer, gout, hypertension, prostate cancer, and rheumatoid arthritis. So we are picking those disease phenotypes based on a relatively wide range of the case disease ratios in the UK Biobank data sets. And also we are specifically picking 2 gender specific disease, which is prostate cancer and the prostate cancer. We want to make sure for those more unique data sets, our method can still have the good performance. So as you can see here, first for each method, we trying to identify the risk regions with either GWAS signals and the TWAS signal. That means like a marginal GWAS test or marginal TWAS test. And based on those potentially risk regions, we further identify the signals that are located in those risk regions to serve as the possibly true signals. So here, as we listed, we can see that although Fabio may not identify the most number of total, signals indicated as the numbers outside of the bracket, but we usually have the highest proportion of the potential true signals among them. So as you can see, also, Fabio usually can identify more so-called true signals against other comparing methods. And afterwards, we want to highlight several examples of candidate causal genes that do have a biological support, and that is also uniquely identified by Fabio compared to the other existing methods. So the first example is called ABCG2. So this is a causal gene we identified for gout. And the biological support here is that there is a study have already resolved that a specific SNP here within ABCG2 was identified to be associated with gout. And also, we have the pathway evidence to support that ABCG2 is a reasonable causal gene for gout. The next example is called SH2B3. So this is a gene uniquely identified in our analysis by Fabio for hypertension. So again, for this gene, it does have GWAS evidence, and also we have the pathway analysis to support how this gene is located in the pathway for this disease, which is more, solid evidence to kind of support the idea that SH2B3 can be a potentially causal gene for hypertension. So as a summary for this part, so we claim that Fabio achieves superior fine mapping resolution, producing candidate gene sets smaller than those from current methods. And also we show that Fabio achieves the highest proportion of risk genes and can also nominate unique disease-associated genes with literature support. So this work has been published in PLOS Genetics in 2024. Sure. Yeah.
Because I'm confused.
Mm-hmm.
If you go back a few slides to where
you
had the, let's see, that one.
This one?
Yeah. If a risk region, you have these 24 risk regions for the different traits.
Mm-hmm.
Now, if there is a risk region that is outside of those 24, for example, for the asthma.
Yeah.
Can you be certain that those are not affiliated with asthma in any way?
Yeah, that's a very good point. So, yeah, so here we are trying to identify those risk regions more possibly to be associated with the trait of asthma here. So, as we indicated here, actually we are using a quite a broader like a criteria, which means we pick a region as risk. Either it has GWAS signals or it has TWAS marginal signals. We will consider it as a risk region. Definitely, there will be other known regions that can be risk for asthma. However, we can also see that for each method of the TWAS-5 mapping, we usually have other signals outside of these identified risk regions. So those signals might be the true signals. However, in this case, it's really hard to tell. So we prefer to use these genes within the risk regions to serve as a true signal here, like the golden standard here as evaluation. Yeah.
And then, further, what do you make of this 228 risk regions for hypertension?
Mm-hmm.
That seems like an awful lot of stuff to have to work with.
Yeah.
And very different from your other traits.
Yeah, exactly.
So, is hypertension actually a trait? Or is it some kind of like a something larger than a trait, something like a condition, or whatever you want to call it?
Yeah, exactly. Yeah, so here because we are focusing on the binary outcomes, and definitely I agree, hypertension is like a very complex disease. It can involving like a very multiple traits, so-called traits, either continuous or binary. So here we were using like the patient-reported outcomes in the UK Biobank data sets. So they do have the report of, like the self-report of whether or not they do have hypertension. So that is the binary outcome we are looking for here. So we just using that arbitrary outcome to serve as a binary outcome for hypertension in our study. But definitely I agree, since there are much more risk regions identified by like simple marginal GWAS and TWAS, we can go deeper for these specific traits in the future. Since this is a method, a method article, we focus more on reporting the method instead of reporting the clinical findings out of the applications. Yeah, so that's a reason here. Yeah, very good point. Thanks. Yeah, I will moving on to the second part of my talk today. So next, I will talk about the MFABIO, which is a multi-tissue TWAS-5 mapping methods. So again, I want to start from the motivations of why we want to develop such a method. So here, as I just mentioned, most of the existing TWAS-5 mapping methods, they rely on single tissue gene expression reference data. So it will cause a problem. For example, it will risk overlooking the true causal genes if the disease-relevant activity occurs in an unanalyzed tissue. For example, for hypertension, since this is a complex disease involving multiple tissues and multiple like parts of the body, so if we only use EQTL panel from for example, from heart, which is less accessible, usually just from whole blood, which is more accessible, we may miss some specific tissue-related genes here if we only use whole blood to serve as an EQTL reference panel. And also, we noticed that so far, only one published method for multi-tissue TWAS-5 mapping, that is called TGFM from Harvard. And that method is still, based on the linear regression model. So that means like other single tissue TWAS-5 mapping method, most of the existing TWAS-5 mapping methods were primarily designed based on the linear regression model, and they were designed specifically for quantitative traits. So again, although linear regression might be an approximation to logistic regression, but we still find that using our probability link might be advanced to have a better, might be better to model the binary outcomes here. So here we present MFABIO. So again, the MFabio is the traditional two-step TWAS file mapping method. So first, we need the EQTL reference panel from different tissues. So here we will generate the predicted gene expression level for each gene and also specifically for each tissue. Therefore, it will leading to a final big matrix, including the predicted gene expression predicted expression level for gene tissue pair instead of just the gene. And afterwards, again, we apply the probability link, which is designed for the binary outcomes to better capture the variance of the gene expression levels among the the more complicated gene tissue structure. So here, I just want to elaborate a little bit more on the probability link here we're using MFabio. So here we apply in the standard probability link between the traditional linear predictor and the binary outcome. And within the linear predictor part, we including both the gene effects terms indicated as G hat beta, and we also including the pleiotropic effects directly from the genotypes. So which is labeled as X alpha here. And we trying to capture both the gene effector size as well as the pleiotropic effects in this modeling for binary outcomes. And also, different from Fabio, we trying to adapt the more advanced and the more popular sum of single effects prior, which is known as SUSY here, to capture the gene tissue pair structure when we considering the EQTL panels from different tissues. So here we apply the standard SUSY framework. We have segmented effects. We have a sum of small single effects and summarized together as the final effects for specific gene tissue pair. And also we leveraging the Bayesian framework. So we have assigned so for each gene tissue pair, we will calculate the final posterior inclusion probability by assuming probability to select each gene. And also we have a conditional probability in addition to that, which means the probability of selected tissue given a gene is already selected. So combine those two probabilities together, we will generate the final probability whether or not a gene tissue pair is selected in our model. So again, we started to evaluate our method using extensive simulations. So since now we do need the EQTL panel across different tissues, so we're using the UK Biobank to serve as a GWAS panel. And however, for the EQTL, we were using the GTEx, which covering multiple different tissues. And also, we same as what I just mentioned for Fabio, we also need to simulate binary outcomes based on the, specifically for the GWAS panel, and the parameters using the simulations are almost the same, especially, except for the number of causal tissues here. We introduced a new parameter as number of causal tissues and to indicate whether or not a method can capture the more complicated case when there are more tissues involved. So here we first showed that MFabio is able to control force signals in the non-simulations. And here I just want to explain a little bit more here. So as you can see, there are only, as I mentioned, there are only one known, published method for multi-tissue TWAS file mapping, which is called TGFM. So we can directly compare MFabio to TGFM. However, there are other single tissue methods, including Fabio and Focus. And also, as I mentioned, there are two more recent advanced methods called GIFT and CTWAS. And we also including those methods. However, for those single tissue methods, we apply them in two different ways. So when there is GT here, that means we just plug in the gene tissue level input matrix for those methods. However, they were not designed to do so because they are not considering the gene expression level correlation across different tissues. And also, we just applying those single tissue method directly for each gene, that for each tissue, and then combine the result all together. So yeah, so that is more naturally way following their original design. So as you can see here, the method, our method MFabio and the method the TGFM always have a better control for signals because they were designed for the structure of gene tissue pairs. However, for those single tissue based methods, it's a bit hard for them to control the false signals because they do not take into consideration the correlations among tissues. So next, we show that MFabio is able to achieve the highest power under different conditions among all the competing methods. So this is under different case control ratios, and this is under different number of causal tissues, and this is under different PBEs and the different PBE2. Yeah, so then again, we step out from the simulations to the real data application. So we just pick the same 6 binary outcomes here from the UK Biobank, and very similar to what I showed earlier for Fabio. We're trying to first identify the risk genes with either GWAS or TWAS evidence to use them to serve as a potentially golden standard to identify whether or not a gene is true or not. And then we trying to identify the total number of genes for each method, as well as the total number of identified genes that located in the risk regions. Yeah, so as you can see here, those single tissue method always have like a potentially higher false possible rate. So they are able to identify more signals and also even more signals located in the risk region. But again, the advantage of our method as well as another multi-tissue method, TGFM, we are able to identify usually more genes with the GWAS or TWAS evidence. And you can see that the proportion of the so-called true signals is much higher compared to the single tissue method. So that was for the gene level. And we also specifically calculate this, generate a result for the gene tissue pair level. So very similar observations. Although single tissue methods can identify more signals in total, but there are usually more true signals for our method and a higher proportion of the so-called true signals. And also, we trying to have identify some more solid gene tissue pairs with literature support. So here we just query the PubMed for publications over the past 10 years for each disease tissue combinations. And we pick the top 3 GTEx tissue categories with the highest publication counts for each disease and consider those gene tissue pairs as the potentially true disease relevant tissues for that specific disease. Again, here, despite of the total number of signals identified across different methods, we calculate the proportion of identified true disease-relevant gene tissue pairs for different methods, and we can see that our method MFabio always achieves the highest proportion. So again, I want to showcase some example of the candidate causal gene tissue pairs that do have further biological support. So the first example is the D2H-GD2 associated with in the lung tissue associated with asthma. So yeah, so it's common sense that lung tissue is probably the most relevant tissue for asthma, and our method is successfully identify the specific gene tissue pair for asthma with biological support. And the next one is the gene CYBRD1 in the breast memory tissue associated with breast cancer. So again, breast cancer might be the top one target for breast cancer, and our method is able to identify the specific gene tissue pair associated with breast cancer. Yeah, so as a summary for the second part of my talk today, so we want to highlight some key features for mFabio. So this method can specifically model the nature of binary outcomes through our probability link with SUSY prior. And also we are able to consider the pleiotropic effects and leverages gene expression reference from multiple tissues. And through the simulations and the real data application, Fabio can identify more evidence-supported genes as well as gene tissue pairs comparing to other existing methods. And also, mFabio is able to prioritize unique disease-associated gene tissue pairs with literature support. So this work is currently under revision. Yeah, so since my talk today is the majority part of my dissertation, I just want to again thank my dissertation committee, and I was co-advised by Dr. Xiang Zhou and Dr. Alex Choi, and I also want to thank all the lab members from Dr. Zhou's lab and also Dr. Choi's lab for this work here. Yeah, thank you all for listening. Yeah, I'm glad to take any questions or comments you may have. Sure. Also work with GWAS statistics instead of individual alike. Yeah, that's a very sharp question. Yeah, that is also a very good point. So the main shortcoming of Fabio, also mFabio, is that since we are using the probability link, or we are not able to intake the individual level GWAS data. We are not able to take the summary level GWAS data. We do need the individual level because we need the individual level data to kind of doing the iteration within the Bayesian framework with probability link. But the good side is that since we are using the individual level GWAS data, we can precisely capture the LD structure. Because usually for when you are using the GWAS summary statistics, you do need the LD reference panel, right? So either you can use the in-sample LD structure, but if the GWAS summary is not available, if the LD structure is not available, pairing with the GWAS summary data, you do need the independent reference panel. So that may cause the mismatch of LD structure. So our method can always capture the most exact LD structure within the data. Yeah.
Yeah. I have a question. I don't know if you covered this, but how does Fabio deal with case control imbalance? Does it account for it appropriately compared to the other models?
Sure.
Degradation,
yeah,
your imbalance case control ratio?
Yeah, good point. So I can quickly show this one. Yeah, actually here on the top left corner, so you can see that Fabio is able, is able to produce quite consistent performance compared to other methods, and also, more better performance. In different case control ratios. So we have the selection ranging from 1:1 to 1:50. So 1:50 is pretty much like the true case in the UK Biobank data set, especially for the six trait we selected here. Yeah.
Why do you because I'm seeing the other methods do degrade a little bit, but Fabio barely degrades at all. So why do you think that is?
Yeah, so that's true. Yeah, so Fabio also degrade a little bit, and I think the major point is that because we are using probability link, so probability link is specially designed for the binary outcomes. However, the other method just use a linear regression as approximation. It's highly possible they lose power when they are approximating. When they are approximating the binaries to treat them as just continuous. Yeah.
For the example gene for the breast cancer, I noticed right next to it is metal 8 that methyltransferase, which is highly implicated in pathogenesis of breast cancer.
Yeah.
So I'm curious in terms of like fine mapping, is that within the range that you think of those as having linkage? And
Yeah.
maybe in individuals, you could look at that sort of breakdown, not just breast cancer, but severity or stage, or maybe there's more structure in the individuals that would help you distinguish where this evidence is coming from.
Yeah, this is a good point. So usually, in addition to the gene tissue pair level like outcome, like the PIP, the probability whether or not this gene tissue pair is selected out, we can also yielded a set of genes, which may including like multiple genes within the same locus here. However, they can be kind of ranked by the probability. As you just mentioned, for example, here, I do not recall the exact statistics here, but CYBID1 might be the most top ranked one with the highest probability, but there might be some other genes nearby with a lower probability, but they are still within the credible confidence set. So we can either report it the whole gene set, or as you suggested, we can do like further studies, or maybe even more clinical studies to make to kind of further prove which one is the true signal compared to the other one, nearby one. Yeah, thanks.